tell you the story of that. I didn't put it in here, but I'm happy to tell that story.
How they picked up on that so quickly, and how then that particular vaccine to do with the adjuvant, wasn't it? Was it the adjuvant issue?
It was a bit inconclusive. It was about a certain brand of vaccine, and they think it's how they were preparing it, how they were splitting the virus. Yeah, that meant it got taken off the shelf.
Back on that safety lecture, how you can be careful about stimulation of the immune system. So, with no further ado,
Thanks, Chris.
Thank you very much.
Are we waiting on anyone else? I think you said there were 7 in 1, 2, 3, 4, 5.
Unfortunately, he's just got a job at Path West, so they refused. They've got a training thing.
Okay.
To change it.
Right.
To change it.
Yep. Okay. My email's there anyway. So this is with this on that gets recorded. Yeah.
Yeah.
Is that
so
okay?
Is it? You just switch the button on.
I've got it's on.
Yeah. All right. Thanks very much.
Thanks, Chris. Thank you. So my name's Leanne, and my email's there. If you do have any questions or anything's not making sense, I'm happy to have a chat and help clarify a few things. I've looked at the whole program that you've got for this particular course, and it's quite comprehensive, and there may be some overlap from different people, but I think it's good to reinforce that to you. But if things don't make sense, just reach out. I'm happy to chat. And even just now, I didn't realize you're quite a small cohort. So let's just, if there's something that's not making sense, just say as we go along. It's probably, other people are probably thinking the same thing. So as Chris said, I did my PhD. I'm from Scotland originally. I did my PhD, so honours in microbiology, PhD in vaccine development. So I worked on a vaccine, pneumococcal vaccine to prevent pneumonia and meningitis. That resulted in a few patents during my PhD, one licensed by Pfizer, and they took that in-house to develop their next generation of pneumococcal vaccines. So that was pretty cool, and that was my first taste of what it's like to be working in vaccine development as an academic, but working with industry. And it's such a massive field, and I was just speaking to Chris there. Obviously, the public's been exposed to what's involved in vaccine development with COVID, and how fast it was, and was everything adhered to? What has to happen at each stage? So I think as a population, world population, we're more informed about vaccines than we ever have been, and about vaccine safety, how vaccines work, and all the different types that there are. So today I've been asked to talk about animal models and their use in vaccine development. So I'll try and put that into place of all the other things you're going to be learning about in this course. And hopefully, vaccine development's quite rigid. There's certain things you have to adhere to, and there's certain safety criteria you have to meet. So it should all make sense when you're hearing this from different people at the different stages of vaccine development, whether that's discovery, preclinical development, or your clinical trials. There's always things we have to adhere to, and we're all working towards that one goal of getting the vaccine into the arms of people. Yeah. So vaccine development takes forever unless you have the whole world working together like we did during COVID. So generally, it's quite a slow process. It's pretty complex. There's lots of different factors going on, and it costs a lot of money. So around about 10 years is what it takes to get a vaccine to market normally, unless the world is throwing money at it like we did during COVID. And not all vaccines will make it to market. So I've got less than 5% on the slide there, but it's probably less than 1%. And that's the ones that make it, they're already promising candidates. They work in the lab, and for whatever reason, they fall over. Too expensive to make, too difficult to make, maybe don't meet safety criteria. And as we go, I always think of a funnel. You have lots of candidates coming in to begin with, and as they funnel down, they keep falling over, and only 1 or 2 make it to the end. And then it costs a lot to develop a vaccine. So that's another reason that vaccine candidates can fall over. You can have the best looking vaccine candidate, can look amazing in all your animal trials, it can look great in clinical trials, but if the cost of making that and getting that into the arms of people doesn't outweigh the cost of what you're trying, if you're trying to prevent a disease or reduce the number of hospitalizations or something, if the cost benefit doesn't weigh up, that vaccine will still not be developed. So there's lots of things that can kill your vaccine as you go along. And I'm a casualty of some of those things. Throughout every stage, safety is paramount. And the reason for that is In other drug development, sometimes people are so sick that you can accept those toxicological effects. So, you know, if you're really dying from something, a drug that could let you have a prolonged life but may have real toxic effects would be tolerated. But for a vaccine, generally you're given that vaccine to healthy people. So the safety profile has to be at the highest standard because you don't want to do any harm to that healthy population. Vaccines undergo a lot of scrutiny. It's not just the scientists, but there's regulatory bodies. So you will have heard of the FDA, for example, yeah? Here in Australia, we have the TGA, Therapeutic Goods Australia, who are constantly reviewing vaccines as they're coming into clinical trials and as they're getting ready to go to market, and then post-vaccine surveillance as well. And Chris Blythe, who I think came and gave a lecture to you out of sequence, I thought he was coming next week, he would have talked a bit about that surveillance and that safety. And I will give you that little story that Chris just pointed to, because I'm not sure Chris did either. But I just wanted to let you know that safety, we don't just wait till we get to human trials to test safety. We also have to test that in animal models to get an indication of how safe it's looking before we move into human trials. Okay, and so the thing that Chris Peacock was just referring to, in 2010, so there's a good vaccine surveillance here in Western Australia. It's one of the best in the world, and it can look for any changes if there's any increase in any reports of something going on. And in 2010, there was a rise in the amount of kids that were having convulsions from a fever after getting the flu vaccine. And it was happening just before the school holidays, and that surveillance system kind of gave a red alert that something's going on. So as a lab scientist, we were able really quickly to get all the different flu vaccines that were being used for kids in Western Australia at the time. We took peripheral blood monocytes, mononuclear cells, PBMCs, that had been isolated from kids. We'd had them stored in our liquid nitrogen for other projects. We got them out of the freezer and we stimulated them with the different flu vaccines that were on the market, and very quickly found that only one brand was given an inflammatory response, so that fever response. So we could measure an increase in interleukin 6 and interleukin 8 that was happening really quickly to this one brand of vaccine. So that formed part of a package of data that got taken to the TGA and said, look, we've tested all these vaccines, this one's given us the same fever response that you're seeing in children, together with lots of information about the vaccines those kids had been given. Quickly put that picture, this has all happened in 1 week, put that picture together that it was one company's vaccine and we needed to stop vaccinating kids with that immediately. So that vaccine got taken off the shelf and is no longer recommended for children under 9 years of age. And then there was a big investigation, what happened, what was going on. It was something to do with how they were manufacturing the vaccine, and they've had to change their protocols and prove safety like never before. So it's just it's a good example of, that was vaccine surveillance and using software to track that, but then you also needed the scientists to come in and actually look at what was happening. It's also a good reason to have samples biobank that we could quickly pull out to start looking at what was going on from an immune perspective. But it's also nice to have been able to provide an answer quite quickly to change that. So safety, I think you're going to hear from Chris a bit more as well, but I just want you to know that safety is not just at the end when the vaccine's being used in kids, it happens throughout. And then you'll probably see figures like this over this course, because everyone's got a figure they like to use to explain vaccine development, okay? And I just like this one because it does show you the pipeline, gives you an idea of how long things take, and then what's involved at every stage. And today, for this lecture, I've been asked to focus on the first two parts, so the discovery phase and then preclinical development. So this is before we've even got it into any human volunteer. It's the part that research scientists are most involved in, but also, like I said, as you're monitoring and evaluating, I have also been involved in looking at how well vaccines are working when they're out in the public. So I've been involved in a a pneumonia vaccine trial in Papua New Guinea and doing surveillance there to see how well that vaccine's working, how we can improve the next generation of vaccine to mean that we protect more children and prevent more deaths from pneumonia. And so as a researcher, I am involved in the whole pipeline, but really where I get really excited is all new candidates. How can we improve vaccines, and how can we get new vaccines for diseases that we don't have any vaccines just now. So we're going to dive into the first part, the discovery phase. And I know that you had a lecture from Alma last week about selection of vaccine candidates. Well, I think you did. That was what was on the program, and she told me she was coming to give you guys a lecture. So I've put her face up so you remember how these all link together. So in the little figure that I used on the page before, it says the discovery phase is about 1 to 5 years. It can be a lot longer. And for example, the vaccine that I'm working on just now, it was a decade of research before we're now making that to go into clinical trial. And at the end of this lecture, I'm going to finish on where I'm at with that. I'm working on a new vaccine. So at that discovery phase, you want to select your candidates, okay? So I've got a slide after this about how you go about selecting them, and the field's forever changing, and as we push the boundaries, we're going to be bringing in things like AI to help with identifying vaccine candidates. What's a mRNA sequence that can be immunogenic and put into a vaccine within weeks. So COVID vaccine development took a year to get from finding the sequence to getting it used throughout the world. It took 65 days to actually get it in the arm of the first person. So it's the quickest vaccine that has been developed. And then I do want to encourage you, it was too much of a diversion to put into this lecture, but CEPI, down the bottom's what that stands for, the Coalition of Epidemic Preparedness and Innovations. It's a big coalition of all the people that work in the vaccine space. They have a challenge. The US government has just put in $5 billion into this program, which is called the 100-day, I think it's called 100-day challenge. But it's basically we have all the technologies in place to make vaccines within 100 days, but it's making sure that everyone's aligned and that when we see the next threat, there's a pipeline that it can go through, whereas with COVID this time, everyone was kind of scrambling around and, they can do that over there, and people were doing things same time, and it was a bit of a scramble. And to be better prepared, I've even read articles about, when we live in Mars, we need to be able to make vaccines against threats, and how are we going to do that, and how basically we can make vaccines synthetically when you have a sequence, so you don't need to be able to grow the infections, but you need to be able to manufacture your vaccine to get it into people. So it's all about how we can embrace technology such as AI to help inform that. But there's a really nice video as well. So if you want to kind of have a little Google of CEPI 100-day, you'll see a nice video around what they're trying to do. So when we find our candidates or antigens that we want to use to get that protection, we need to show that they're immunogenic, okay, so that we can get some kind of protective response. Normally that's an antibody response because antibodies give you long-term protection, but it doesn't have to be. It can be another immune response, and the nasal spray that I'm working on developing is actually an innate immune response that you get that gives you short-term protection, and then we think that can actually train your innate immune response. So when immunogenicity, normally think of antibodies, but just to be aware, there can be other ways that you can elicit protection. And then you have to show efficacy, so how well does that immune response protect? So there's different models you can use to show that, and it's very specific to the infection or the disease that you're trying to prevent. And then, as I said, safety. And your models that you use can be different. So your efficacy model is normally an infection model where you challenge. Safety could be that you give a really high dose of the vaccine and you look for what response you're going to get, and you have parameters. Obviously, you can't kill people when you give them the vaccine. You don't want them to get really sick. There's parameters like how much of a reaction is acceptable for you to then take that further and into human trials. So I'm not sure the content of Alma's lecture around vaccine candidate selection. This is just a little figure showing all the different types, it's not all of them, the main types of vaccines that can be developed. So, you know, you can take your whole virus and you can attenuate it in some way. So you can genetically modify it, or you can split it up, or you can take a strain that's not very good at causing disease, so like your polio vaccines, and use it that way in like a whole virus. You can inactivate, so you can kill the virus and give a killed virus or a killed bacteria. And then you've got your nucleic acids, so your mRNA and DNA vaccines. Most vaccines that we receive are those little green dots, so a subunit vaccine, and that's when we purify a protein or a polysaccharide. Actually, polysaccharide's a carbohydrate vaccine. So you purify something, normally from your bacteria, and that's what gets injected in. It's a much quicker pathway, development pathway using that process, because it's what we do all the time. But the downside is often your vaccines aren't very immunogenic, or they only protect against a subset of strains. A good example is the pneumococcal vaccine, I'll talk a bit more about in a while. But the pneumococcal vaccine, there's 100 different types of pneumococcus, and the first vaccines that came out protected against 7 types. And then obviously bacteria are way more clever than us, and they just evolved and changed their polysaccharide jackets. And we then had disease from non-vaccine types. The next generation of vaccine comes out, it can protect against 13 of the 100 different types. We saw a little shift, but really the bacteria mutated again, could change their polysaccharide coat, and then other species could come in as well. And now we're up to 20 different pneumococcal types, but again, there's 100 different types of pneumococcus. So it's kind of, you're not really changing much. You're probably removing some really troublesome types, but you're not getting to the core of the problem. And for example, the surveillance work we did in Papua New Guinea, when we swabbed the noses of kids, we found 64 different types of pneumococcus in their nose. Some kids carrying up to 5 different types at one time. So if you've got a vaccine just now, they only have the 13-valent vaccine, so you can tell that's not really going to have an effect on pneumonia in that population. And then we can improve vaccines by conjugating them, so taking the subunits and sticking them to other things that make them more immunogenic, and that works better in young kids. And then we can take a toxin and we can make a toxoid. So toxins, normally if you have antibodies to that toxin, you will be protected. Obviously you don't want to inject a toxin into anyone's arm. It's not going to have great effect. But if you make a toxoid, so you can change the toxin so it's no longer toxic. That was actually my PhD. I took pneumolysine, a toxin from pneumococcus, and genetically mutated that into a toxoid that retained its immunogenicity but didn't have the negative side effects. So I kind of just want you to get from that, there's lots of ways to make a vaccine, and these are all the different ways we've been making them to date. And you know, there's been a lot in the literature just now about mRNA vaccines, so when you can synthetically take a sequence, that speeds up how quickly you can make your vaccine. So once you've got your candidate selected, you do 2 tests, and I've just linked that. So the 1, 2, and 3 up there demonstrate immunogenicity is number 2. I've got some more examples in the next slide, but normally this is done in mice, and normally you will give your vaccine and then you will give a booster, and then you'll measure the antibodies, or you'll measure some other output such as T cell responses or B cells. And I did see you have a lab demonstration about ELISpots, where you actually get to see the B cell, like, it's pretty cool, but you'll see little dots, and that's the B cells making your specific antigen. So that's another way we can test that we've got an immunogenic protein or other vaccine candidate. And then number 3 down the bottom would be when you introduce a challenge. So number 2 is just checking, if I give you the thing that I think is going to be a good vaccine, will I see an antibody response, or will I see some type of immune response? And then number 3, if I give this vaccine and then I challenge with the thing I'm trying to prevent disease from, will I see protection? So basically, if it's a lethal infection and you've got a vaccinated population, do they survive? And I'll give you some examples of what that data looks like as well, some real life examples. So in the past, when we've taken things like polio vaccines and we've inactivated the polio virus, that was kind of a look-see, like let's take the infection and what's causing the infection and do something to it so we can put it back into people. We've become a bit more advanced in how we develop vaccines, and this is called the rational vaccine design process. And so then that's to begin with, rather than just looking at the infectious agent and working out how can we use parts of that to protect, we go back, so at the very beginning of this picture, to the infected person and are actually able to look at, okay, what's happening there? What do they have lots of antibody to? If I take serum from them, and if I put that, if I was to quote, say you take the virus and you take their antibodies, does that then neutralize? I don't know if you've heard of neutralizing assays. That was a big thing with COVID. Did you make neutralizing antibodies? Could your antibodies prevent the virus from causing infection in you? So if we look at all that, that gives us lots of information as to what's the most important proteins or the key epitopes, and then that lets us kind of quickly rationalize which antigens we should be selecting. So then that lets us, I've got it here, epitopes that have that neutralizing activity. It kind of, we know a lot more with protein structure now as well, so I don't know if anyone loves structural biology, but learning, how a protein folds and which parts are on the outside, which are the epitopes that an antibody is going to see, and we can predict quite well now, it's called epitope mapping, but we can predict which parts of that protein are on the outside and which ones are going to have a high affinity with antibodies as well. So then you can say, actually, it's this sequence, and I know the RNA sequence for that, or the DNA sequence, and I know it's going to fold this way, so then we'll get these neutralizing antibodies, so this is the area we need to focus on. So that's really speeded up how we make our vaccines, but also we always have to come back and test them in our animal models, show that we're getting that immune response in that animal model, and then allow us to move on to our human trials. I've already told you about COVID time. Okay, so last year, at the end of last year, there was lots of headlines. There was a law passed in the US saying that animals no longer needed to be used for drug development. So this course that you're being taught about is all drugs, and then you have vaccines within that, okay? The vaccine guidelines are a lot stricter, and you can't avoid using animal models. So although the US came out and passed this law, FDA then has to follow and say animals are no longer needed for drug development. This doesn't include vaccines, okay? We still need to have that animal testing before we move into human trials. And there are other models that we can use, and we're always trying to reduce and refine when we are using animal models. We don't take it lightly, and there's never any superfluous use of animals. There's lots of good lab assays, using cell cultures or maybe organoids, that we can test some toxicity or potency of your vaccine. And there's also some in silico modeling, so using computer models to predict things. But ultimately, it still needs to be tested in an animal. And I want you to know I don't take that lightly. I am actually super scared of mice and rats. I can't touch them. I've tried my hardest. I have the nightmares I had during my PhD when I had to be in the animal house doing those experiments have scarred me for life. But it's still something that my team needs to do, and I have amazing people in my team that are able to do those animal experiments for me. But it's something, it's something I carry, and I just want to acknowledge that not everyone feels comfortable with animal testing. But also, I don't know how many people would like to have a vaccine that hadn't been tested in an animal beforehand. And cosmetics and industry and all that's a very different thing, I feel, to vaccines to prevent infectious diseases. That's me, by the way. So this is just some examples of the animal models that would be used for vaccine development. These are all the models that get used for influenza vaccine development, and they're not always suitable, and it depends what you're planning on doing. Most models will start using a mouse, and on the next slide I've got the pros and cons of different animal models, but generally mice are easy to handle, well characterized. There's lots of reagents we can buy to test antibody responses and the like, but sometimes they're not very reflective of the disease. And so, for example, for influenza vaccine design, ferrets are a very good model, and they're very good for transmission modeling. So mice don't really transmit flu to each other, whereas ferrets do. So if you want to check, does your vaccine prevent transmission of flu to another person, which is kind of the holy grail for vaccines. So the first vaccines and the first COVID vaccines, they've protected us from dying, but they haven't stopped us transmitting the disease to other people. And it's a next level, and it's a whole other type of immune response you need to prevent transmission. So the models that you need to use are different depending on what you're looking at. Guinea pigs actually give a very good febrile response. So if you're looking at diseases that cause a fever, they can be a better, more sensitive model to use than your mice. And then also, the mice we're using are inbred as well, so you're not really getting a very reflective response, but you're getting something that biologically isn't all over the place. So you can kind of analyze your data and be confident in your findings. There's not many pig models, but it depends what you're looking at, and it depends what vaccine you're developing. And then there is a move away from using monkeys for vaccine research. So they get called NHPs, so non-human primates. And normally it's a small group size tested at the very end. We can't do any of that here in WA. We have no approvals to do that. And it depends on if it's a completely new infection, a completely new way of developing a vaccine, whether you need to go into those primate models. And there's very tight regulation. And the FDA has also said we want researchers to answer the whole vaccine space to try and get away from using primates if you can. So for example, the vaccine that I'm developing, I only need to show protection into animals, and they can be small rodents, which is enough for us to get approval to move into human trials. But it depends what you're working with, depends on what models you would need to use. Okay, these are just the pros and cons on the most common animal models that are used. So as I said, it's easy to use animals, it's easy to order them in, it's easy to breed them in-house. When we talk about reagents, so that's if I want to look at the antibodies that have been made to my vaccine, I then need to get some kind of tagged antibody against that mouse antibody. So it will be anti-mouse IgG, and it will have some color that I can detect by FACS or by ELISA or some way. So we'll have a look when you're doing your ELISpot assay, the anti-sera that you're using. I don't know if you get to see the actual original vial or if it gets all aliquoted out, but that will be raised either in cells against humans or in animals. But there's so many reagents out there that we can purchase for mouse work. And then obviously there's lots of knockout mice as well, so you can start looking at immune signaling in different pathways. Like I said, mice tend not to get that fever response, and there can be quite a lot of variation, not just within the strain of mouse, but the substrain. And we've actually had a whole 2 years of our work not going very well because of genetic drift of the strain of mouse we're using. We're now using something different to what we had at the beginning, and it's become a bit of a nightmare. So using guinea pigs can be a bit more relevant to disease, but they're bigger, they're harder to handle, smaller numbers at a time. They cost a lot. There's not that many reagents developed for using them, and there's not that many strains available. And then also, using monkeys ethically, I don't think I could continue developing the vaccine I'm developing if that's where we needed to go, because for me, I really struggle with that. But it is an important part. For example, Ebola vaccine development, primates are the key animal to test that in, especially as that's a disease we think's crossed over from monkeys into humans. And then also it's very expensive, and there's not that many places in the world that you can do those experiments. So lots of regulation and permission and real justification on why you would need to use them. Don't worry about everything that's here. I just want you to get a sense of the shape of these timelines, okay? So these are all just examples of when you're designing an immunogenicity experiment, what they kind of look like, okay? So the three on the top, I just want you to get a feel of. Basically, normally you start in your mouse with your vaccine candidate. You will give your vaccine to the mouse, so let's look at the top left one just now. And then you'll give it 1 or 2 boosters. So the first picture, the mouse is getting 2 boosters at week 3 and week 6. And we can take blood samples throughout. So that's how we measure the immune response. Normally that's from a tail bleed, so we can take, it's kind of like taking blood from your arm here, but taking it from, they have a vein down their tail that's easy to get the blood from, and that's where we can measure their antibody response. There'll be an endpoint to the experiment where other tissues can be collected, like spleen or lungs, or it depends what you're looking at. So top ones are you're just checking you've got an immune response, and then the ones down the bottom are where you have some kind of challenge. So the one on the left, there's 3 doses of immunization, so they get their primary and then 2 boosters, and then challenged with whatever your disease X is, yeah? And then they will be monitored. So you'll have a group that get placebo, and then you'll have a group that get your vaccine at a minimum, and then so vaccinated, placebo, challenged, and then you monitor their survival. And I've got some graphs to show you what that would look like. And then the figure on the very bottom right-hand side that has an A next to it, that's our challenge model that we're doing just now with the vaccine that we're developing. So I will tell you more about that story at the end. It's called Spritz-OM. So we give that, and then we then have, it's a model of ear infections. So it's a virus that drives a bacterial ear infection. So we give 2 challenges, flu and then non-typable Haemophilus influenzae. And then we collect tissues to see is there less of the bacteria or antivirus when you're vaccinated compared with your placebo. So I don't want you don't need to memorize these things. It's just to get an idea of what those timelines look like. So they're normally weeks apart, can be days apart, doesn't always have to be an intramuscular injection, can be intranasal, can be oral. So there's, how you design your experiment depends on what you're developing. Is it an oral vaccine? Is it a systemic injectable vaccine? So there's lots of ways to design it. And this is just an example. Don't stress about what all the data says, but it's just an example of then what would that data look like once you've measured that immune response. So you've got your mouse, you collected blood before you started to know what the baseline was. You've given them, in this case, you've given 1 + 1, so a prime dose and then a booster, and then you wait a few days and then collect blood again, okay? And then down the bottom is just the data from this particular immunogenicity test. And what they were testing really is so VLP is your virus-like particle, and then they've just added 2 different antigens. So an antigen gives you an even better immune response. It kind of just gives a little bit of a boost. So plus alum and plus Adjuvax, 2 different adjuvants, and they've measured all IgG and then breaking that down into different subgroups. And basically, if I was to pick, I'd probably say the pink or the blue is looking better than the virus-like particle on its own. And then obviously your controls, you wouldn't be expecting them to make any antibodies. They wouldn't have any IgG to your target. So these are 2 examples of efficacy data. So the one I showed you before was just, are you getting an immune response? This one is, does it work? If I challenge the animal, am I going to see any protection? And I've chosen these 2 because these are the 2 case studies that I'm going to share with you in a minute. So GPM Vaccines is an Adelaide-based company. I just want you to look at survival time. So basically, they have a challenge model that if the mouse is not protected with your vaccine, it dies. That's quite a severe model, but obviously they're wanting to protect against death from pneumococcus. PCV13 is that 13-valent vaccine that's on the market just now that I told you about. Only protects against 13 of the 100 different types. So that's your kind of, is it better or is it the same as the one we have on the market just now? And then that's their two different formulations in the yellow and in the blue. So basically they've increased the survival time and it's looking as good, if not better, than the vaccine we have on the market. And then this is just from my team. With ours, we're looking at the number of mice that have an ear infection. So when we've given them Spritz OM, we have less than 20% of mice are developing an ear infection in comparison with the control. So that's what you'd be expecting your efficacy data to look like in your first animal experiments. So then the yellow part was the discovery. Takes ages, but it's quite exciting. And you know, when you find your vaccine candidate, it's an exciting space to be in. And then the second part would be your preclinical development phase. It's really rigorous and it's highly regulated. And I've only got one slide on this really because we just basically have to follow the rules. So the first part is making sure that once you've shown that it's feasible, you know you have an antigen, it's immunogenic, and it looks like it's given protection against the disease you designed it to, you then want to go on and show how would you make this vaccine. So you want to make that under GMP. I don't know if you've heard of GMP before, but good manufacturing practice. So it basically needs to be made at a certain facility before it can go into humans. That's the stage we're at just now with our vaccine. It costs $1 bazillion, so you know, we're raising the money to do that just now. We have already spent $500,000 and we've raised another $2 million to do that. And then once you've shown that you have a good way of manufacturing your vaccine, that it's, you know, it's not just done in your lab and you know, you've got all the right paperwork and accreditation, it can then be released and you can test the toxicity. So this normally gets done externally, so we are paying a lab to do this for us over East. And this can also get called a toxicology study. And so this is certain tests that are done. You go up to the maximum dose you would ever want to use in humans. So you have that information. It's normally done in rats. You know, they'll look for fever responses. Is there any toxicology happening? Look at all the different tissues and lots of microscopy to find out if there's anything that is a cause for concern. If that's all within the parameters that are acceptable, you can then apply to use this in a human trial. So you apply to your regulatory body. So in our case, that would be the TGA if we're going to be doing our trial here in Australia, the FDA if you're in the US, the EMA, the European Medical Association if you're in Europe, and if you don't come under of any of them, nationally there's a advisory committee that you can apply to, and then you also need to get ethics approval from a human research ethics committee. So those are very standardized. There's only one way to do it, and you just have to follow the rules. So just to finish off, I've got two case studies, and the first one I'm going to tell you about is a company that has spun out of the University of Adelaide. So I think it's good for you to realise that vaccine development at the early stages often comes out of university or a research institute, and then evolves into that industry space, and that can happen. Like I said at the beginning, Pfizer licensed my patent, so they basically could take all my ideas and my data. They then owned everything, and then they run with it. Or you can spin out as your own company, which is what we're looking to do with our vaccine candidate just now. And this is what these guys have done. So it came out of University of Adelaide, and they spun out into this GPN Vaccines, also based in Adelaide. They focused on pneumococcal disease. So if you're not familiar with the pneumococcus, many of us carry it in the nose. Lots of kids carry the pneumococcus in their nose and have no symptoms and pass it to each other very happily. However, the pneumococcus can cause meningitis, pneumonia, and sepsis. And it's one of the major killers of kids under 5 throughout the world. And like I told you, the vaccine that we have just now is less than ideal. So this is Pfizer's PCV13 vaccine. They take the sugar capsule of the pneumococcus, so it's polysaccharide capsule, and have conjugated it to proteins to make it immunogenic in children. But they've done that for 13 of the 100 types. And like I said, we're up to 20. There's a 24 valent coming on the market. There will never be a 100 valent. This is the most expensive vaccine we have on the market. It's $50 per dose, and kids need 3 doses. And we are looking at reducing that, so maybe 2 doses. And in developing countries, there are agreements where they can make their own vaccine for their own population, so that brings down the cost as well. But still very expensive. And if we were to put all 100 types into that, the vaccine would be like this size to get into someone's arm. Like, we're never going to have 100 types. So this company have said, well, how can we get away from using the polysaccharides? Let's get something that's protective across all pneumococcus. And that's been the holy grail for the last 30 years. So they take the streptococcus and they irradiate it. So just like we do with fruit and veg, so that it lasts longer on our shelves. So I've just a little picture of when you irradiate strawberries and they stay nice compared to when you don't. It sterilizes the outside. So you can, it basically kills the bacteria that are on your fruit and veg and help them last. So all your apples that are in the shops for ages have all been gamma irradiated. They have gamma irradiated pneumococcus. It allows all the proteins on the outside to stay all in their natural form, so they stay immunogenic, but the bacteria cannot replicate. So you're not giving an infection to someone, but you're giving something very similar. So it's a dead product. So, you know, I showed you the antigens you could use. This would be given a dead whole cell bacteria. Again, I don't want you to stress about the data, but it's more just showing their publications where they've proven that they have safety and immunogenicity in an animal model. So the little gamma, the radiation sign, is that's their product in comparison with a placebo control. So they get increased survival time. In the graph on the right-hand side, they have more IgA and they have more IgG, and these are just survival plots here. So basically the mice that have the square, that have been given the vaccine, they survive and survive for longer in comparison with the control. So that was enough for them to say, okay, we've got our immunogenicity data, we've got our efficacy data. This has just continued. They've gone and tested against lots of different types of the pneumococcus to show that it's not just specific to one type, because again, they want to have that cross protection. I just want you to get used to seeing the shapes of the graphs. Don't stress about the detail, but it's those survival plots, and it's just given that confidence that you've got something that's worth taking into humans. Again, they've done a lot on their showing their safety. It's just a snapshot here. Safety, immunogenicity, and testing, and they've just finished their phase 1 trial. So that's to look at safety in humans. I believe you have a lecture after me that's about phase 1 and phase 2 trials. So hopefully this takes you on to what happens at those phase 1 and phase 2 trials. And then just to finish off in the last few minutes, this is what I'm working on just now. So Spritz-OM, we're using whole cell bacteria as well. This time we're using a live commensal, so beneficiary bacteria that's found in the nose of children that don't get lots of ear infections. And the reason we're doing that, ear infections are the main reason that a child will see a GP. They're the main reason that they get given antibiotics, and the main reason that they will have surgery. There's millions of cases throughout the world every year, cost $5 billion a year in the US alone to treat. A burden throughout the world, and First Nation people throughout the world are at the highest risk. And then kids that don't have their ear infections treated can have lots of hearing loss and speech delays, and it can have a real impact on their education and their outcomes for life. And we and others have shown that non-typable Haemophilus influenzae causes the most amount of ear infections, so that's why we focused on making a vaccine for that. And there's no NTHI vaccines on the market just now. So Spritz-OM is using a friendly bacteria called Haemophilus hemolyticus, and we spray it up the nose. This is giving you an example of my team's data. So on the very left, you've seen that already. So the number of mice that get ear infections significantly reduced when we give them the Spritz-OM up the nose. So this is just one dose up the nose. When we measure how much of that bacterial pathogen can get into the ear, 99.99 reduction in percent reduction in the amount of bacteria in the ears of mice that have had that spray up the nose. And then this is just measuring their weight, so the aqua green line in comparison with the placebo, you can see that their weight returns back to normal more quickly than the placebo group. So that's enough for us to take this into clinical trials, and this is just a little snapshot of me and where we're at just now. So, you know, it's been a decade of getting to this stage. We've got our preclinical data. We're now working with a Belgian partner to manufacture it, a Portuguese partner to package it up, to send it back to us to do our phase 1 trial, because there's nowhere in Australia that can do that for us, and then also test the toxicology in those standardized experiments, so we get approval to move into our phase 1 trials, which we will be doing at Linear, and I believe you guys have a trip to Linear as well, so that will be cool for you to see where things like this happen. And then obviously we move through those really standardized phase 1, phase 2, phase 3 to get it onto market, and you can see down the bottom how much that roughly is going to cost us, and yeah, why we need to be fundraising as we go and look for investing partners. So with that, I'm just going to finish, and this is a thanks to my team. So we're based over at Telethon Kids Institute, and this is my awesome team that helped me do all those experiments and lots more. And thanks to you guys. I'm very happy to take any questions, or you're welcome to email questions. Want to work in vaccine development?
You say you wanted to spin off into your own company. How does that work with taking the work from Telethon?
Yep. So it's a very good question. So just now, all of my intellectual property, so my patent, is owned by Telethon Kids Institute as an employee there. They have to release that patent into a spin-out, but they would be a main, they'd have the major equity share because they have backed all that, you know, and they're basically a partner for life as we spin out. And for them, you need to spin out or license with someone for this to become a product. Telethon Kids cannot manufacture a vaccine and have it on licensed for use. So it's part of what has to happen, and they're very supportive of that. I'm quite lucky in that all of the IP is within our one institute. If you have, you know, it's you and someone else and you're in two different places, then you have to have your agreement, and then it splits it between different institutes, and you know that can be not just 2 institutes but 3 or 4, and then that can get a bit messy. But it's actively encouraged, and at Telethon Kids they've spun out 7 companies, and it's something that they, you know needs to happen. Otherwise this won't go anywhere. And you know, if it's not me pushing this, I don't know who else would. So yeah, but watch the space. In the next 6 months, we've said, based on our data, we may be ready to spin out or not. Yeah. And then I have to balance that as an academic with a research team that's doing lots of things versus a business, and you know, what's my role in that? So I could be a chief scientific officer or chief technical officer rather than the CEO running all the business sides. But then when you're early startup, you've not got money to have a massive team, and it's your baby, so maybe you are the CEO to begin with, and you have a board around you that advise you. And but it's a really exciting space, and I feel in Australia the biotech space and the early startups around biotech is really it's growing, and it's going to get bigger and better. Brisbane's actually had a lot more investment than we have in WA, but that's happening here just now, and I can see that we'll be like Brisbane is in maybe 10 years' time. So there's momentum, and Australia's a very it's well known worldwide for doing clinical trials. So that bit before of not having to go overseas to make it or to do your testing, if you can keep that all here, then that boosts that pipeline as well. So it's definitely a growing and exciting space to be part of. And there's quite a lot of biodesign courses, and there's a lot of activity here that if you are interested, you would find there's seminars, there's how to grow your business, how to spin out, all those things. You'd be able to to go along to those things. MTP Connect is something to look up, and that's government funding to help boost biotech innovation here in WA and connect us throughout Australia. Well, like I said, any other questions or anything, happy for to take them at any time. And good luck with the rest of the course. My pleasure. Do you guys have another lecture straight after? Well, do you
I just thought I looked like I knew what I was doing.
Is it recording?
Oh, okay. Just do it very gently. don't. It is working. Recording goes. Hopefully it will work, but I think there might be. Oh, that's working now. Okay. And then there's a point. It's a great pleasure to introduce Simon Scott, who is the co-director of Neuroscientific Biopharmaceutical.
Well, just the protein.
And
It's present throughout the body, but very present in neurons, so has
targeting
neuroprotective.
neuroprotective. Actually develop new therapeutics for it, I imagine.
Yeah.
And prior to that, he was at Linear
Yeah.
Research. So yeah.
It's been a while since I've given a lecture, so but please jump in with questions as we go along. So hopefully it'll be a bit more interactive than just me talking. It's now 20 years ago I was in these halls here doing my undergrad in microbiology, which doesn't seem like it was really that long ago, but and then after with healthy volunteers running clinical trials, and I think it's great that it's being brought into curriculum now, because I left university, I didn't even know what a clinical trial was. So Charlie's Hospital over in C Block, working in osteoporosis trials, but I didn't really love it. It wasn't really anywhere to go in the position. Studies all over the place. So yeah, I went in there and as a project manager, and then sort of went into doing startup and ethics work, and then operations management, business. Biopharmaceuticals, or NSB for short. But yeah, so we have one lead candidate that we're hoping to get into clinic this year, but we've had creates a tetramer, but it's really difficult to manufacture, it's difficult to assay, it's difficult to put into a formulation that you can give to humans. It comes with a lot of problems. So along the way, so that's how I ended up here. It was a bit of a winding journey that, well, it really started in a pub in London when I was working. was all right. I was just giving some background, so
Oh, okay.
we hadn't got into the topic yet.
Hopefully it will last. I'll just leave it there, just so I have a backup.
So the topic is sort of where we're talking about focusing on vaccine trials and what does phase 1 and phase 2 of vaccine trials mean. I think you went into it to just the previous and I guess what are you trying to learn from, what indication of efficacy or effectiveness can you get from these early stage trials? So hopefully we'll get phase 1. This is your first trial in a human for a new biomedical intervention. You're doing it usually in a small group of people, and these have usually when we move on to phase 2, so we're looking at a large group of people, could be several hundred, even up to 1,000, depending on the design of the study. And you're evaluating further safety through, usually you're dosing for quite a bit longer in phase 2 than phase 1's very short-term dosing. That's in short of a vaccine trial. So vaccine trial, similarly, you might have a few more participants, but I've done a lot of trials where we're dosing over 100 participants under a phase 1. Design of the study, which we'll go into a bit more. Phase 2, so the key difference here with a normal clinical trial is that because vaccines as well, so it kind of brings that in, whereas clinical trials you're looking at people with disease, and you'd always be looking at an adult population at that, really in phase 2. Cool, so we'll run through this quickly because it probably covers a bit of what you just learnt in your previous lecture. But made to a quality standard that's good enough to be put to humans. You've done a bunch of safety studies in today, so that's sort of the ability of a foreign substance, such as the antigen in your vaccine, to provoke an immune response. So it's going to have you got enough to show that it's safe, and have you got enough to show that it should hopefully do what it's supposed to do, and has it been manufactured? I talk about more clinical trials because it's something I've been dealing with recently. But and you also might introduce the idea of challenge studies, where so if you can if you can get all that done and you can get a Human Research Ethics Committee to sign off on it, then you're ready to go into your phase 1. So I'm going to go kind of a fair bit into that occur in vaccine trials as well as regular clinical trials, is that these studies were normally designed so that they're trial to receive a particular treatment. They'll be double blind, so the staff working at the site, who, you know, the doctors, the nurses, we do also have this idea of an unblinded team who will know, and they're usually involved with the actual giving of the vaccines. And these trials will also so this one reads, the slide design came out a bit weird, so it kind of goes from right around. So most vaccine trials will follow a pretty similar visit schedule. So after you've been on a vaccine trial, this might actually be done on the day of vaccination. It just depends on what assessments they need to do. Generally, if they require you to have blood tests, the doctor, they'll run you through the consent form. You ask any questions, then you're consented. Then you'll have a bunch of procedures. They'll do a physical exam. You might have or you progress on to being dosed. So whether that's on the same day or a different day, so say it's a different day, you come in. Pieces, and then the doctor will go back through everything and check that you are continue to be eligible for the study, at which point they will sign you off to be randomised onto a treatment. The dose, come back into the clinic, give you that vaccination. Like 99 times out of 100, a vaccine is going to be and that would usually, the length of that will depend on what stage of development they are. So this is sort of the first in human one. They might want you to stick around for 4 to 6. So we keep them in for a little while, but during that time we're giving, so what's important during these trials is that you'll receive a temperature probe. So you go home and you'll take your temperature every day and write in your diary to say, you might be given a ruler as well. So still written down on paper, but it's very slowly moving towards electronic records and things. And so you might be on a study where you get an e-diary and you get an app, so you just log it in all. Usually on day 7, you'd come back in for a safety follow-up, and that's when you'd complete your diary. So sometimes with the paper one, you'd blood pressure, check your temperature, make sure you're all okay. Also, what I didn't mention is that pre-dose before your vaccination, they'll take a bunch of bloods. You take bloods, and they'll so look at how are you producing antibodies to the based on the come in day 1 and then say day 22. It might be 4 weeks apart, it might be 2 weeks. It might be 3-dose regimen I've seen in some, so it just depends on the study design. Placebo one. Depends on the randomisation or study design. During this time as well, they're also collecting any adverse events that occur. What is important for the participant to do is to record everything that happens. So even if you've got a headache because you went out. While you're on that study, but with the vaccine study, because we're going to follow someone up for a year, we don't want every headache you've ever. What we call serious adverse events. So that's, if you're hospitalised, if you had a life-threatening occurrence, death is one as well. But obviously. Say all the COVID-19 studies, you wanted to know if a participant contracted COVID at any point during the study, even if they only had mild symptoms, because it was important. Collecting bloods from you, but they're usually about a year long, and then you finish your participation. To wait till a year to get paid, no one would continue in the study. So you set up payments to keep people engaged. Not that participants are only doing it for the money. So can you give this vaccine in escalating, so dose, we're going to start with a small dose and we're going to keep increasing till we're in a range where we think we're going to tolerability, and we're comparing that against the placebo participants who would be involved. And then our secondary objective is to look at this develop the vaccine for. All right, so if you want to participate in one of these phase 1 vaccine trials, if you're a female, you need to be not pregnant and on adequate contraception, generally because at that stage they just haven't done reproductive. And also that generally your female partner is also on contraception, because we want to make really sure that, because nobody vaccinations, just because it might interfere with what they can pick up in the blood. So there might be a 28-day restriction on any other vaccinations. What they're looking at, they might have travel restrictions. So they might say, for the period of study, you need to not go to Southeast Asia for that time. So just it's usually going to require some preparation, because it's all going to be in different parts. They're not going to make it up in set doses or with set They generally be in the fridge. They might be in a freezer if they don't have stability data to support refrigerated storage, which can happen in these earlier. That participant has been randomised, so doctors sign them off, they're good to receive it. All their details will be put into a computerised system, which will spit out to site, and that typically happens because at that stage, when all the components are put together, they don't know how long it's going to last for and still be viable. And then they dose participants right then, so they can do it in a short period of time. What happens then, that team that do the dosing, they then can't be involved in any other part of the study. So because there's a lot of involved with a phase 1 trial, it's a group. For a tour, you get to see it, it's a lot different to when I started there. It was pretty much empty. Now it's packed to the rafters. Um, also can call in a code blue medical emergency if there's an anaphylactic reaction and things start to go south really quickly. So you need access to that. You need specialist equipment. Who are just doing this day in, day out. So because of these reasons, it sort of makes it impractical to do it in a sort of through a GP clinic or phase 1 sites around Australia. So there's about 5 in Australia that run them. In which case you employ a contract research organisation. So just from a phase 1 perspective, I just wanted to run through sort of a couple of real world examples of studies that I was personally involved in, just to give Merck Sharp and Dohme, one of the biggest pharma companies in the world, and they were looking at a vaccine for dengue fever, which still unmet need, still no one's cracked that one. So it's at day 0, then month 1 and month 2. Participants came in for their vaccination. One site. So because Merck were Merck and they have a lot of rules, they decided that our nursing team on site couldn't make up the doses. So we used the hospital pharmacy, but because they didn't have so much stability information on their got phoned down to the pharmacy, they'd randomise them, and then they'd contact, we had a couple of unblinded staff members to say, do not enter unblinded tasks going on. So the lab staff had to stop what they were doing, leave, then they'd take the vials out. Going on, the unblinded team had to fake going in and getting a vial out and putting it in Esky and taking it down. So it was all just to maintain this blindedness. Only really a dedicated unit that could do the kind of thing. A more recent one, so not long after. Their one was actually, so whereas your Moderna and your Pfizer are the mRNA base that's coding for that spike protein. we got, because of the way they designed the study was so fast paced that we're like, it's never going to work in a two-side approach because we're going to have to move really quickly and dose. $18 and $60, and then 60 above the age of 60. And we did that sort of, I think there were sort of cohorts of 10. So you'd bring enroll the next lot of younger participants. At the same time, we'd enroll a cohort of older participants at the previous dose, and we kind of kept going in this. Ethics, so the ethics committee Linear uses is a centralised one based in Adelaide called Belbury, and they were willing to take sort of a more pragmatic approach to review. On what they'd done in that time to continue to allow the approval, so we sort of had a rolling approval of the study. So that was all happening while we're rapidly enrolling all these. We wanted to go on and do more of an expanded study and bring more people onto it, but the Chinese government decided that any vaccines developed in China and we'll go and sort of, part of my finish off is, I guess, to sort of discuss how COVID really changed the whole landscape, like how we talk about vaccines. Phase 2 vaccine trials, so you've successfully completed your phase 1, you're ready to move into phase 2. So a couple of little notes on this. So move into phase 2. In this study, you're looking at least sort of 200 participants. You may see up to 1,000. Little cohort of patients. 2A, 2B. So 2B kind of maybe you're blurring the line with phase 3 and you're trying to get more of an efficacy read. Based on your phase 1 data, but you'd still have the placebo-controlled group. You're going to be reducing how much safety. There's not really, and there's no benefit from really running a phase 2 for any longer than you did for a phase 1, because you're really just collecting these immunogenicity samples through. What's your regimen going to be? Is it a 2 vaccine approach? 1? Do you want them 3 weeks, 4 weeks apart? You want to nail all that down in phase 2. At least enough participants so that you can have some statistical significance to get meaningful outcomes on your safety, immunogenicity, and In a phase 2 vaccine trial, so as I mentioned previously, you might be including we've had big issue with RSV this winter. The US got hit really hard with it last year, but there's 2 groups really that get affected by RSV. So it's really, you want to introduce these at-risk groups at this point. You usually have the requirement to be healthy is cholesterol, and they're on drugs for everything. We also introduced this concept of assent. So any participant in a clinical trial who's an adult needs to say that you understand what's happening. So it's not formally legally binding like the consent form is, but it's just that there's a record that this child it might still just be all the different vials, but we're probably going to have a reduced selection, so we might only have a couple of doses, 1 adjuvant kind of thing. So it's a bit more. In it that you just draw up. If it's a pre-filled syringe, you might get a kit that has, say, so if it's a 2-dose regimen, you might have 3 syringes in there in case. Administer it and then remain blinded, so it reduces that requirement to have additional staff who can't do other tasks on the study. Does mean that because you've got like less of these really strict requirements, you might be able to branch out who runs your trials. So you may still get they can do the recruitment, they can dose the participants, they can collect samples and store them and ship them. So they've got those basic facilities, but not to the standard. Is that the CRO, the organisation that's managing the sites, they need to make sure that whoever's running it can run it to a standard that we call GCP. Stringent again, but you'd be unlikely to bring them in at phase 2 just probably for those reasons. So this database, so all your information goes onto the database so that it's all pooled, all the data, and that CRO will send out what we call a clinical study monitor. A data management group will clean all that data to make sure it's nice and clean before the stats group get it, and then they can really go into it. So yeah, so that CRO is really, at that point they're really taking over management, whereas at phase 1, a site like Linear has a lot of say in how things where they do challenges. So very few places do these, particularly depending on whatever the pathogen is. We don't currently have it. The latest seasonal flu strain, flu vaccine, or placebo, and then they'll be put into a locked isolation room where without having to, dose thousands of people and look at how many on placebo versus how many on active, were protected against whatever the disease. But it depends on the nature of the pathogen, whether you need a full PC3 isolation facility or if it's Linear recently did a study with wait until they developed clinical symptoms of the sore throat and other pharyngitis and stuff. So then they would treat them with oral. But no one's ever bothered to check what dose of penicillin is effective. You just give them a big dose. But so TKI are actually trying to do something about that. But and it's all effectiveness in a phase 1 or 2 vaccine trial. So so efficacy is this idea of this is the ability. Reducing symptoms, it's reducing hospitalisations, it's saving lives. So there was a bit of a stigma about it, but I guess it's important to know vaccines aren't always going to. So you can't really formally assess that until you've got the thousands and thousands of participants you need in phase. You would not go to any regions where you get dengue, then you're not likely to get cases during that time. So that's when you're reliant on the blood samples. Second one, are you getting more antibodies or is it the same? Like what's happening in that blood? And you can tell a lot from blood, and that's what you're really looking at this. So the blood is everything in these trials. So that's why you need a phase 1 site who know how. As it's required to be done. So when we talk about, so we know that everyone knows, COVID-19 really. but Pfizer BioNTech and Moderna, and then to a lesser extent Novavax, who were a bit later, but they did it seemingly with all of it. Conspiracy theorists who might say that they just cut a lot of corners, didn't do everything they were supposed to do. They were putting out vaccines that weren't safe, but that wasn't the case. They're rolling into phase 2, phase 3. It's all happening at the same time. So they took on an incredible amount of financial risk to really, because most decisions as you're going through your development process, whereas they just had to go, we're going all in, which for Pfizer, who have very, very on it, and as I put there, Pfizer sold $37.8 billion worth of their vaccine in 2022. And if we can make a vaccine within 12 months for COVID, what about the next pandemic, epidemic? Expectation is now that you'll do it. With this initiative, they call it a moonshot, is essentially to say for any disease we should be able to make a vaccine within 100 days. And it's increased awareness. People probably didn't know so much about clinical trials, or everyone wasn't an expert on them in the early days. But cool. So that's just a recap of the objectives. So I think we went through when we're talking phase 1 and phase 2. Currently working and where more of my expertise is, but I have done a fair few vaccine studies, so hopefully it was vaccine trial looks like, but hopefully gives you a bit of an idea of how you would go about designing one in a typical sense. And then yes.
I have got a two-part question.
Two-part. Okay.
First of all, how much would a principal get paid for this? And then is there also like a caveat you could
Can't pay participants for risk, unfortunately. No. But so it's usually your time. So if you're doing, which we'll go into the next lecture, if you're doing your time and you're willing to commit to coming for these visits for a year, so you might, I think they were probably paying around sort of 18 to 2. J365, and how many you can do depends on, so that while you're in that vaccine trial, you can't be part of another trial. Day 7, your next trial might just have a criteria to say, oh, you can't have participated in a clinical trial for the last 30 days. So then cool.
Was that COVID trial that you did, the
Mhm.
vaccine trial? Did
The Pfizer and Moderna ones were approved because you couldn't stop participants taking, it would be unethical. What their antibody response was versus the other ones, so it didn't impact on the validity of their data. So yeah, it became a whole thing. I didn't think I could talk for that long. But there you go.